All right, let's get started after that little technical disaster. I feel like I was on an ad for an antiperspirant or something. Okay, last time we discussed neighborhood effects on health and the distinction between compositional and contextual effects. We talked about the importance of physical, social, and sort of the medical service aspects of place. And we considered specifically or especially the spatial distribution of death during the Chicago heat wave and the results from the Moving to Opportunities experiment and the Broken Windows field experiment. Today I'm going to be emphasizing how social factors may affect our biology at a much deeper level than we've so far been seeing. What do we know exactly about how the social becomes biological at the genetic level? And this, in fact, is a growing cutting-edge field. And we've seen many examples in the class of how social factors of all kinds, sort of education, income, poverty, social support, income inequality in the area that you live in, and so forth, can affect your health, and specifically proximate health measures like your blood pressure, your heart attack risk, or your immune system function. The question remains, could something still more profound be going on? So let's consider some of the ways today that social factors could actually interact with your genome, not just the sort of physiologic expression that your body might manifest, but actually the kind of the ways in which your genome is regulated. We're going to consider all three possibilities today. One is a set of ideas regarding evolution and sociocultural change, how the social world around us might modify our evolution as a species. A second idea has to do with gene by environment, or specifically social environmental effects, how social environment might modify or regulate the expression of our genes or interact with the expression of our genes. And lastly, we'll consider sort of a very nascent and interesting field, at least to me, of social epigenetics, how social factors might regulate the expression of our genes. Now, ordinarily, humans are seen as evolving under the pressure of the environment they face over vast timescales. And this environment may be seen to be composed of three different types. And most of you, any of you who've had exposure to evolutionary biology, are familiar with the notion that species evolve under the pressure of the environment that they face. And this environment could include physical features of the environment, the sunlight, the temperature, the humidity, the salinity of the water, the availability of water, et cetera, the altitude of the creature, and so forth. Or it could include, very obviously, biological aspects of the environment. What are the predators or prey that the organism is facing, or the pathogens that the organism is confronting? And lastly, and most interestingly for today's purpose, it actually could include the social environment. Who or what are the other conspecifics that surround the individual members of a particular species? And in fact, the social environment has often been neglected as an evolutionary force. And yet social factors are crucial, especially in our own evolution. And they may, in fact, have led us to be intelligent in the first place. And there's a set of ideas in evolutionary biology at the moment known as the social brain hypothesis. And the social brain hypothesis posits that one of the reasons we are intelligent is, in fact, the necessity not to make tools, but to make friends. The whole reason all of us are sitting and having this conversation today may have to do with the fact that we had to constantly interact with other members of our own species. And in fact, one of the greatest predators that humans typically have faced over evolutionary time has been other humans. The main thing that has killed us is other people, not other creatures or even environmental constraints. So something about the existence of those other people and our ability to cooperate with them or get along with them has, it is argued, affected our capacity for intelligence and our capacity and a whole suite of features that we have, physical and neurological features that we have, that affect our ability and our interest in interacting with others. So this theory stresses the special challenges posed by living in close proximity with others of one's own species and confronting the complex demands of a complex social environment, not a complex physical or biological environment so much. An environment that involves constant cooperation and competition. But what's interesting about the social environment, much more than the physical or the biological environment, is that at least until recently, we humans, both individually and collectively, could only create and modify our social environment. So unlike the physical and biological environment, which, for millennia, for 10 eons, we've been unable to do anything about that environment, it just impinged on us, the social environment is something we've always created. And the argument is that across evolutionary time, we've created this social environment, and it's fed back and acted as a kind of selection pressure upon us, modifying the expression or modifying the fitness advantage of different genes within us. So this environment can feed back and affect our evolution and the expression and function of our genes. Now, in fact, it's also possible to imagine that we humans may have been changing our social environment in ways that affects our genes not only over long-term horizons, but even over historical time periods. So when you think about some of the biological effects that we are discussing today, or some of the ways in which our social exposures could feed back and modify or regulate the expression of our genes, you have to think in terms of very long, hundreds of thousands of years, medium, sort of thousands of years, and even very short, in ways I'll show you very soon, decades, tens of years, time spans that could affect our exposures and our experiences. And if we grant the relevance and salience of our social environment, and if we see the social environment as malleable over historical time, it begs the question about whether we are actually evolving in real time in observable ways in response to social changes we are both genetically and non-genetically compelled to make. Now, as we've been discussing in the course for the whole semester so far, in some ways there's nothing new about this claim, because our biology and our culture have always been in a kind of interesting conversation. For example, rising socioeconomic status with industrial development resulted in people becoming taller and living longer, which are biological effects of a cultural development. We invent industry, we become richer, and therefore our bodies change. We become taller and we live longer. And conversely, taller people require a change in architecture. So if any of you, raise your hands if you've ever been to sort of a medieval home, for example, or a colonial home. And have you marveled at the small size of the beds or the low height of the ceilings? Because you feel so tall in that kind of a building. So as our bodies have changed, we've had the kind of biological change, it's fed back to a suite of cultural changes, for example, in the case of architecture, the kind of buildings that we build. So here, however, we're talking about something more fundamental than we've been talking in the course so far. Here we've been talking not about sort of the length of our life or our height or those types of physiologic changes. Rather, we're talking about our actual genome. Could our genes be in conversation with our cultural products over time intervals such as those that are shown here? Now, the best example we have so far is the evolution of lactose tolerance in adults, for which there were a number of readings assigned for today. Now, the ability of adults to digest lactose, raise your hands if you can drink milk comfortably and eat cheese and so forth. Exactly. But the ability to do that only confers evolutionary advantages when there's a stable supply, in adulthood, when there's a stable supply of milk available, such as after milk-producing animals such as sheep, cattle, or goats have been domesticated. And in fact, the advantages are several. Why would it be? It's obvious why babies should be able to digest lactose, because they drink their mother's milk, but why should you guys be able to digest lactose? You're not drinking your mother's milk anymore. So what would be the advantages of that? Well, it's very useful to be able to drink milk. It's a valuable source of energy. It's a source of necessary hydration during times when the water is spoiled, and confers a number of advantages. But you could only benefit from those advantages after humans had done something in historical time, done something, namely domesticate animals. And amazingly, several adaptive mutations have occurred in widely separated populations in Africa and Europe just over the past 3,000 to 9,000 years. So just in kind of dim history, over a historical time period, we have done this thing, domesticate animals, changed our environment, and in changing our environment have resulted in all of you being able to digest milk, which you would not have been able to do 20,000 years ago, prior to our domestication. And these mutations, the ability to digest lactose as an adult, are principally seen in populations of people who are herders and not in nearby populations who have retained a hunter-gatherer lifestyle. And this is also an example of convergent evolution in response to the social environment, because there have been multiple different mutations that have arisen that give rise to the same phenotype, namely the ability to digest lactose when you're an adult. And in fact, there are many similar sorts of examples that we can begin to think about as well. other kinds of things, other kinds of social and cultural rules that we've created over historical time, which have changed our selection landscape and modified our genes in the kinds of allelic variants, the kinds of genes that you have now compared to what you would have had, absent these developments. So changes in population density, how, when we urbanize or congregate in particular ways, development of agriculture, the migration to high altitudes, rules regarding endogamy, when we have religions that specify who you can and cannot marry. Those types of rules, it turns out, affect the genetic landscape that we all have. Family size, desire, to reproductive size, or in fact, even the introduction or the invention of modern medicine. I would argue in 1,000 years, people will look back and say, or even in 500 years, I think, frankly, we're getting more, this is a crazy idea, but I think it's possibly true. I think we're getting more myopic because of medieval lens grinders. I think we invent the ability to create lenses 500 years ago, and as a result, I would have been eaten by lions, 20,000 years ago. I can't see a damn thing without my glasses. But now I've survived and I've reproduced and I have more kids and they're going to be equally myopic. And you can imagine over historical time that you're going to have the same kinds of phenomena. So how can some of these things play out? Well, a similar story to the lactase persistence that I told you about a moment ago can be told regarding relatively recent mutations that confer advantages in terms of surviving epidemic diseases such as typhoid in Europe. These diseases, these epidemics, were made more likely when agriculture was invented and could support a higher density of human settlement, and when far-flung trade became possible. So we go from living in small, isolated bands to large population congregations and movement across great distances of other human beings. This is all the culture, the civilization we've created over the last 10,000 years. But that sets the stage for the emergence of new epidemics, which in turn cull the population of individuals with certain variants and not other variants. For example, I don't know if it was in the readings, but cystic fibrosis. If you're heterozygous for the cystic fibrosis gene, it is felt there's now increasing evidence that conferred in it a relative resistance to typhoid. And so the reason we have this thing nowadays, the reason some kids are born with CF, is in fact because we survived the epidemics from Europe, which epidemics arose in historical time because of our invention of cities and trade and so forth. So there's another example of how culture can change our genes. And these stimuli can be technological, as in animal domestication or medicine, or social, as in terms of population density or family size. And in fact, other arguments and still other papers that I didn't give you make the claim that agriculture, the invention of agriculture, has affected the biology of malaria, which in turn has affected the prevalence of at least two different low-activity versions of alleles for G6PD, or glucose 6-phosphate dehydrogenase, between about 1,600 and 12,000 years ago. And still another example has to do with the Tibetans who split off from the Han Chinese and settled high altitudes. This is like a response to a variety of sociocultural phenomena occurring in historical time. This population moves to high altitude, and they have a profile of hemoglobin expression well suited to high altitude elevation, dissimilar from those of us, like the Greeks, who have spent the last 10,000 years at sea level. And in fact, increasing population size alone that we saw at the beginning of the course when we looked at the explosion of the human population, over the last 100,000, 10,000 years. We were stable for eons of time. The population on the planet was stable. Increasing population size alone may have accelerated genetic change, because more people means more possible new adaptive mutations. And the agricultural revolution fostered larger tolerable population size and hence fed back into more genetic evolution. And indeed, there's evidence that as many as 1,800 out of the 22,000 human genes are actively being selected for in historical time, and some of these stimuli, so we know that about 10% of the human genes are changing or have evidence in ways I won't go into today, have evidence that they're under active selection over the last 3 to 30,000 years. And it cannot be the case that all of that is simply due to changes in our physical and biological environment. Probably some of that change has to do with the social environment, which we, of course, have been making for ourselves. And this is a map taken from the readings of phenotype and genotype proportions in different population groups from the relevant parts of Africa. On the left is sort of a phenotypic map, and I forgot right now, I can look. The LP indicates lactase persistence, the LIP indicates lactase intermediate persistence, and the LNP indicates lactase non-persistence. So you can look at the persistence of lactase, the phenotype, on the left, this is the relevant part of Africa. It's probably faint, the Africa map. These are the countries, Sudan, Kenya, and Tanzania. And here, mapped in the same sort of population segments, is the relevant genotypes. And the pie charts are the approximate geographic, the pie charts are located in the approximate geographic locations of the populations, and there's a correspondence between phenotypes, genotypes, and herding behaviors seen in these populations, which is what the argument is based on. And another similar argument has to do with rules and practices and circumstances favoring endogamy, which is another social factor that can affect our genome. This shows the global distribution of marriages between couples that were related as second cousins or closer, namely had an F that was greater than or equal to 0.0156. So this is the fraction of people of marriages that are consanguineous, such that the individuals within the marriage are first or second cousins. So they're closer than second cousins. And if you look at this around the world, you see that the world varies in the proportion of individuals of marriages which are consanguineous to this extent. And worldwide, couples related closer than second cousins and their progeny account for about 10.4% of the global population. And in fact, mortality in first cousins, it's much less clear about second cousins, mortality in first cousins, progeny of first cousins, is maybe 3.5% higher. It's not huge. It's not like if first cousins mate, or frankly even siblings mate, that the children will necessarily die, but it is the case that consanguineous marriages confer some elevated risk in mortality and in genetic deficiency in the offspring. Now, therefore, a decline, and keep in mind as well that for tens of thousands of our years of our prehistory, we didn't move very far. Most of the time, you only one you could marry or mate with were your second cousins or your first cousins, right? You didn't, like in this melting pot that you guys are, find people that are completely different than you. Most of the time, people interacted with their relatives. But there's been a decline in consanguineous marriage over historical time, and this is expected to reduce the prevalence of complex genetically based diseases. And there's several contemporary social processes that are affecting and mainly reducing the prevalence of consanguineous marriages. So again, population density, when you live in more dense situations, you've got more choices of sexual partners or marital partners. Group size, migration, the fact that we migrate over the surface of the earth, urbanization, smaller family sizes, which reduce the number of related individuals in each subsequent generation. All of these can affect the availability of unrelated partners. And so, frankly, do norms regarding marriage. We can invent rules about who it is or is not permissible to marry. If you've ever taken an anthropology class, you surely have read about such rules. We can invent those rules, and in inventing those rules and applying them, modify, for example, a number of phenomena at the genetic level. For example, we can the rules that we invent and certain other processes can affect levels of homozygosity in the genome. So if two related individuals mate, longer fractions of the genome are more likely to be homozygous for whatever trait or whatever allele is present at each locus on the genome. And in fact, levels of homozygosity are very similar in Native Americans and in Middle Easterners. So if we go and we look at Native Americans and we look at their genome, we see that it's very typical for Native Americans to have the same variant of a given gene on both strands of the DNA. Now, why might that be, on both chromosomes? Why might that be if you're a Native American? Anyone taking evolutionary biology or? So because of the founder effect. So there were a few original Native Americans who migrated across the Bering Straits. We think maybe 2,000 or 3,000 people. And they came across the Bering Straits, and all the subsequent offspring were a product of unions among those individuals with that gene pool to work with, okay? So there's a population bottleneck. Imagine in the limit that you had two people, let's say two unrelated people, or maybe even they were siblings, it doesn't matter, and they go to an island and they start reproducing, and the only people on that island that ever arise are offspring of those original founders. You can probably have the intuition that there's limited genetic variation in the founders, and that over as the population reproduce, it'll be like having like a strain of white rats. Eventually they'll be homozygous at every locus. Well, it turns out that among Middle Eastern populations, the level of homozygosity is the same as that seen in Native Americans. But the Middle Eastern populations never had a population bottleneck. They had rules about marriage. They said you can only marry individuals who are within your religion. And so those social rules had the same impact on homozygosity in these populations as a population bottleneck created by having to cross the Bering Strait however many tens of thousands of years ago. So here again is a social factor that can have a very profound impact on the nature of the genetic constitution that we and other individuals have. So that's the first story. How different sorts of social things across long historical epochs, thousands or tens of thousands of years, certainly hundreds of thousands of years, how different kinds of social things can affect the variety of genes or the kinds of genes or the allelic variants that we have as a species today. Let's turn to the second example, which was also given to you in your readings. This is the so-called gene by environment interaction effects. And here the idea is not that the nature of the genes changes, the same genes. Now the idea is that the social environment regulates or intersects with, affects whether particular genes that individuals have find an expression or are useful or are translated into a phenotype. So the idea is that whether a gene expresses itself depends on the environment and specifically the social environment. And the very famous paper about this, which is still constantly being re-examined, was in your reading and demonstrates what we're talking about. So the promoter activity of the 5-HTT gene, which is in the serotonin pathway, the promoter gene for that, the promoter set region for that gene can be modified. And the short S allele of the promoter region is associated with lower transcriptional efficiency compared to the long allele. So if you have the short allele, you don't get as much expression of this gene than if you have the long allele. And on the left shows the probability of having a major depression episode according to the number of stressful life events, according to whether you're homozygous for the short or the long version of the promoter region, or you're heterozygous. And what it shows is that if you have if you're homozygous for the long, pretty much no matter how many stressful life events you have, the probability of becoming depressed is roughly constant. It does rise a little, but for simplicity, assume it's flat. Whereas if you have the short, the more stressful life events you have, the more and more likely you are to express depression. So whether or not you become depressed in response to a stressful life event may depend on what gene variant you have. And conversely, whether the gene variant is relevant, this is the important part, whether the gene variant is relevant to your experience of depression depends on whether or not you experience stress. If you never experience any stress, you might never have reason to express that gene. And that's shown here on the right-hand side, the probability of major depressive episode according to whether the children were treated, no maltreatment, probable maltreatment, or severe maltreatment, again shows a similar picture as shown on the left-hand side. So if you have the S allele, you are more sensitive to stressful life events, but this only mattered if you were actually exposed to such events. If you were not exposed to such events, you were indistinguishable from individuals who had the L allele. Childhood stress could predict, and on the right are results of analyses estimating the association between childhood maltreatment, which took place between the ages of 3 and 11 years, and adult depression, which took place when the adults were between 18 and 26, as a function of the 5-HTT genotype. Childhood stress predicted adult depression only among individuals carrying the S allele, not among the LL homozygotes. Is that clear? Another example. And finally, for our third topic of the three I discussed at the beginning, of how it might be that social factors could drill down to the genetic level and have something to do with the nature of genes we have or their expression, and occur over very short time periods, might be ways in which the social environment doesn't change our genome or doesn't moderate the expression of our genome or its effect, but rather affects the actual expression of our genes. The regulation of the expression of our genes and variation across individuals in how they are expressed falls to a set of processes known as epigenetic effects. And these, in turn, may be under social influence. And there are two main categories of epigenetic regulation of genes: the methylation of DNA strands and the acetylation and other modification of histone proteins. So now the question is, okay, how can social exposures change which genes in your body are turned on and off? And recent work is opening up still more remarkable ways that our social environment may be in conversation with our genes and begging what I think are some of the most exciting questions at the intersection of the natural and the social sciences today. Our exposure to factors such as these, such as poverty, starvation, prenatal care or abuse, family size, the language volume and complexity that children are exposed to in youth, or education that they receive, or in other work that we've done, we've shown that people go through critical periods in their lives. Neonatal period is obviously a critical period. The other period is the period of time when adults reach sexual maturation. And there's a demographic phenomenon known as the operational sex ratio. Raise your hands if you've heard this. The operational sex ratio. The operational sex ratio is a very interesting idea. It's the ratio of sexually ready females or males to sexually ready females. And if you're reaching sexual maturity and you're looking for partners, what you really care about is not the size of the population, and not even the size of, but the size of the opposite sex of the population, and specifically the size of the opposite sex population that's available to you for reproduction. And we did some work that looked at how the male to female ratio, when human beings reach, when their age is 18 to 22, how that affects their longevity. And for example, we found using two very different data sets, including one data set of high schoolers in Wisconsin, we found that the male to female ratio in your high school graduating class, the higher it was, the less long the men lived. Raise your hands if you went to an all-boys school. That's very bad, because there were no girls to be had. So that actually turns out that going to the higher the ratio of boys to girls for boys in such high schools, the worse it was for the boys. Now, in that paper, we didn't show the mechanism of this effect, but it's likely that there were ways in which that was a stressful experience for boys, and similar things happen to women, incidentally, that it was a stressful experience, and that changed the regulation of sex hormones in us, stress hormones in us, and changed the expression, the control of the genes that guide the expression of those hormones in ways that are quasi-permanent. Anyway, so these types of effects, poverty, starvation, parental care, family size, language volume, education, sex ratio that the organism faces, all of these things can modify the expression of our genes in possibly adaptive ways. And what is more, and this is a crucial idea, these epigenetic processes may leave long-term marks. A child raised in poverty or subject to abuse may be marked for life, their body and their genes even shaped by the experience of these types of stressors. So the argument here is that if you're a new baby and you're born into an environment that has not much food, or constant warfare, or violence, or abuse, or neglect, that your body says this is a dangerous environment, and it upregulates certain genes, which make perfect biological sense, and downregulates other genes, but it doesn't just do it temporarily, it affects the way your body matures. It rewires your brain. It affects which parts of your body get more or less blood supply. Your pancreas might grow and your brain might get smaller, or vice versa. I don't mean that literally, but approximately. And so as a result, you inherit that body, which then stays with you for the rest of your life. So these critical periods may mark individuals. And these sets of ideas may in turn, to be very clear, I'm not saying this is the only explanation for the intergenerational transmission of things like child abuse or poverty, for example, but it may play a role. Children that are raised by parents who abuse them may have different sorts of bodies, different epigenetic markers, which in turn then are transmitted to their offspring in ways that you can read about or that I'll touch on in just a moment. So some key experiments, and incidentally, this is all speculative with respect to humans. It's logical. We have very limited evidence in humans, but we have increasing evidence in rodents. And some of the key experiments have been done by Meany and Moshe Ziff at McGill University and other labs around the country using rats. Now, a rat's maternal behavior may be quantified in part by the extent to which it licks and it grooms its pups. So if you're trying to understand what's a good rat mother is one which licks and grooms its pups, and which makes it easy for the pups to drink milk by arching its back in a way that exposes the nipples and makes it very easy for the little pups to get the milk that they need. And so this is sort of a rat's behaving in this sort of way, grooming and taking care of their babies. And rat moms, it turns out, vary in the extent to which they evince this licking and grooming behavior. And it's sort of roughly normally distributed. Some of them are lousy, they are sort of neglect their pups, and some of them are very, very nice, and there's an average sort of pup rat mom behavior over here. And in fact, you can actually practice this yourself if you want. You can practice being a rat mom. You can go to this website and get a little avatar, a little rat avatar. And I think if you Google lick my rats or something, lick your rats. If you Google that, there's a website in Utah, and it goes lick a rat pup, and then you can start licking the pup and taking care of it, and it's like a little game you can play, and you can see what happens to your pups according to, don't do that now if you don't mind. You can sort of see what happens. So you can practice being a rat mom and watching the epigenetic effects on your offspring. And what happens in this case is the following. As this maternal care that the moms might evince upon the babies affects the regulation of the glucocorticoid pathway, which we discussed earlier briefly in the course, in the offspring of the moms. For example, rats that neglect their young and do not lick or groom them result in the pups having increased hippocampal glucocorticoid receptor expression. Gene promoter regions for the glucocorticoid gene are methylated more or less permanently and differently under the two conditions, affecting the amount of glucocorticoid receptor expression in the pups as a result of the social exposure of the pups. So what happens here is a mom that has low licking and grooming. She's neglecting her pups. The promoter region for the glucocorticoid receptor gets methylated, which reduces the expression of this gene. You get decoyed glucocorticoid receptor expression. So when the receptor declines, what does your body do in response? It starts pumping up more and more of the stress hormone because it's not having the effect. You get high corticosterone levels, high anxiety in the offspring, and they in turn go on to evince low licking and grooming to their offspring, the second generation. So you can begin to have a kind of Lamarckian transmission of acquired traits in these types of animals via this type of epigenetic control. Conversely, over here, you do not have the methylation, so you do not, the promoter protein is able to bind to the receptor. You get increased expression of glucocorticoid, get low corticosterone levels, low anxiety, and high licking and grooming of those offspring in the future. And other labs have gone on to generalize these experiments to look at not just maternal offspring behavior, so how the mother treats the pups, but different kinds of social environments. So raising the rats in more collective versus less collective, large versus small groups, how to sort of social exposures like that modify these and other similar pathways. And in fact, there's changes in basic brain neuroanatomy have also been found in these rats. So it's not just that these pups temporarily, these methyl tags basically stay there for the rest of the life of these pups. And for the rest of the life, based on what the exposure they had as they were young, affects whether the glucocorticoid receptor is expressed or not, the extent to which it's expressed. But also, their brains develop in response to this in different ways that are also permanent. So you get a different brain according to whether your mother treated you well or not if you're a rat. And cross-fostering experiments have shown that these effects are not genetic. So if we randomly assign and we take some of these pups and we give them to this mom, and she takes care of them, and some of these and give them to that mom, we can show it's not genetic inheritance, but epigenetic control that's causing these effects. And in humans, some initial work validates these ideas. For instance, a study of suicide victims who had suffered abuse, suicide victims who had not suffered abuse, and controls who died but not by suicide shows similar patterns to these. Abused victims, suicide victims, had much lower glucocorticoid receptor expression than non-abused suicide victims or controls. And so this raises two important points. First, we carry with us biologically records of the world we experienced before. And second, we can actually transmit this. If you were raised in a famine or under a violent state or were abused, your body may change in ways that affect how you raise or are capable of raising your own children. And in fact, this makes sense if you think about it. The epigenome, this whole set of processes, is a biological mechanism that serves as a medium for adapting the genome to altered environments it may face during life. Our species and many other species have evolved the capacity to say, okay, you've got a set of genes, how should you express them depending on the world you face? It makes sense that an organism would be able to do this. If you're born into a world of plenty, you upregulate or express certain genes. If you're born into a world of paucity, you need a different kind of body to survive in that world. And if your early life experience is one of hunger and danger, you need different kind of body to cope with it than if you had satiety and safety. Oh no, that's terrible. And also, did I blow by? Oh yeah, I never explained this slide. So I got so excited I didn't explain it. So here, what's happening is so here are the two strands. Here's your chromosome. You guys probably all learned this in high school biology. There's a strand of DNA which is coiled around these little histone proteins, and then in turn the histone proteins can uncoil and reveal the strands of DNA, which can be in red transcribed. And here's a sort of cartoon example of a methyl group which can bind in certain locations. Here's some methyl groups, and the binding on the ladder of the DNA affects the ability of the proteins of the DNA to be read. And independent of that is a process whereby you can acetylate the actual histone proteins, and that acetylation can affect the expression of genes that are within the coils contained within the histones. And there's a third mechanism of epigenetic control, which I didn't mention and won't mention, which has to do with microRNA and RNA silencing. Anyway, the thing that the Meany and Ziff experiments were just looking at methylation. And what you can maybe even see in this terribly pixelated image, this shows the length of dendrites in the hippocampus is related to the treatment in first postnatal week. So if the rat had low licking and grooming, and this is a slice of brain tissue, and they've stained particular in the hippocampus, and they stained particular neurons and looked, here you can, even in this terribly pixelated image, you can see that the high licking and grooming has a very different and more elaborate set of neuronal connections than on the left, and I need to fix this image. And here's some sort of data that from the same paper, I think Meany in Annual Review of Neuroscience 2001, looks at how offspring in high-licking grooming mothers have increased hippocampal glucocorticoid receptors. So here's different cell fields, different regions, or different ways of looking at the hippocampus, and here's the number of grains, way of counting the expression of glucocorticoid receptors, and the high versus low in each of the different cell fields are always better, have more expression. And the similar results here are shown using glucocorticoid receptor mRNA, looking at low licking and grooming, and higher means more. So we can find both physiologic and anatomic effects as a result of this maternal behavior. So, in a field that we and others are beginning to calling biosocial science, a crucial and challenging topic of research in coming years will be the following. How and why does the social become biological? How do we literally come to embody the social world around us? I've been waving my hands a lot this semester saying, well, social factors can kill you. And they can kill you. I hope you believe me by now. But the question is, how exactly do they kill you? How exactly does poverty kill individuals? What happens in the body at the biological level as a result of these social exposures? How does social support buffer those stresses? What exactly does it do in your body? And we are beginning to go down the causal chain from the exposure to kind of neurological, psychological, and physiological effects, further and further down eventually to the cellular and subcellular level. How and why, in fact, do cultural and social forces reshape our genome over our lifetime and over evolution? And these questions are both hard and important in my judgment. They're hard because they're very technically demanding. It's not easy to do this kind of interdisciplinary research. It requires a set of skills that span the social and the natural sciences. And it's not enough, just if you're good at genetics, you might not be very good at measuring social exposures or knowing which ones are important, or vice versa. If you're really good at understanding social exposures, you might have no clue as to how to go about disarticulating or understanding which layers or which types of epigenetic control might be most important. It's technical, it's demanding, and it's important because it can occur at multiple levels, as I've already given you a kind of hint today. This is an intellectually very interesting and difficult field, understanding how all these things might work. Of course, there's a problem of bidirectional causation. I've alluded to that a little bit already today. So this is where experiments are extremely helpful in teasing it out. And not surprisingly, it's of course morally fraught. And for reasons which I had an argument once with a colleague of mine in the sociology department here. And I was making the claim, which I take as self-evident, and some of you in this room may not, I take as self-evident that there's an incredibly important role of biology in human behavior. That biology plays a really important role, not only in your individual behavior, but actually in our collective behavior. That we can understand many aspects of our social lives by understanding our biology better. I'm not a genetic determinist. I do not think that's the only thing that explains human behavior, but I think it's just sheer ignorance to deny the role of our genes in social processes. So I was having this argument, very heated, with one of my colleagues, and he said, absolutely not, there's no role of biology in culture or in social life. And I looked at him, I couldn't believe it. I said, well, what about violence? 90% of the murders are done by guys. I mean, they have a Y chromosome. Do you think that might play any role? And he said, no. Now, I just find that just like head in the sand kind of ignorance. So I'm not saying I'm not saying that, this is it. Like, I'm going to finish a course in sociology and tell you it's all about the biology, because that's totally false as well. But I would be remiss if I suggested to you that if we just understand poverty or had interventions that or understood social factors alone, we would have a complete understanding of the phenomena that are at least of most interest to me. And they're morally fraught because as soon as you begin talking about these things, people worry about eugenics, and there's a very nasty history of the exploitation of genetic explanations to sort of rate people of different kinds into different categories, which is of course not at all what I'm talking about or what I'm interested in. Now, it may even turn out that they're morally fraught for a final reason, and then I'll close. Human virtues, like love or justice, as I talked about when we were talking about networks, human virtues or cooperation or altruism or kindness that sustain our sociality or that relate to our propensity to be just or to cooperate might themselves even be involved in a sort of large-scale feedback loop, such that we come to internalize the goodness in the world deep within our genes. These sort of values that we take as intrinsic or as important or even as self-evident that philosophers try very hard to explain may actually be values that structure the social world around us and that we came to internalize all the way down to the genetic level, such that we have a kind of genes, on average, which predispose us to be cooperative or to live in groups or to act in certain ways towards each other. And this is an additional and final reason why these things are so morally fraught, because it hints that some part of what we take to be the right and good in the world actually has to do with our genes. Because maybe these virtues determine our genes at least as much as our genes determine these virtues. Thank you. See you next time.